Hi, everyone. I'm Matt. I finally made it here. Don't fly through Chicago ever if you can help it. Yeah. Um, so it's my great pleasure to welcome our keynote speaker for today. Ting just got promoted to associate professor at University of Washington, so congratulations. Um, So she works in clinical trials, causal inference, and multimodal AI for biomedical science. Her work supported by an NIH R35 Mira grant and a PCORI grant, among several other sources of funding. And in addition to being a great scientist, she's also a great mentor. So four of her students were recently awarded American Statistical Association Student Paper Awards. And she's developed widely used R packages, but hasn't submitted anything to Bioconductor yet. So I think our goal today should be to convince her of the merits of submitting a Bioconductor package. So on that note, happy to welcome our speaker.
Thank you so much, Matt, for the kind introduction. And thanks everyone for being here. It's really a great honor for me to present here. Today, I'd like to talk about essentially a story, which is from association to causation, genetic anchored causal inference in human biology. So this talk combines several works of our group recently, and hope this is interesting to you. So I'll start with just three questions, and my talk will be organized around these three questions. The first is, what can genotype tell us that just observational studies and planned association cannot? The second is, can genetic anchoring answer complicated questions rather than just effect of one exposure on one outcome? And the third is, can we forecast what a drug will do before the phase 3 trial using genetics? So let's start with this background. And this is a very expensive failure in 2006. So this is a Illuminate trial that enrolled actually 15,000 patients and then assigned patients to torcetrapib on top of this standard of care, torvastatin. And then this torcetrapib is developed to inhibit CETP, which is a well-known target for change cholesterol. And the torcetrapib actually did exactly what it was designed to do. So it increased HDL cholesterol by 72% and decreased LDL cholesterol by 25% in this trial. However, the trial terminated at a very long follow-up. It had a median follow-up of 550 days. So it's a long trial, and with so many patients and so long of duration, you can imagine how expensive that was. However, the trial was terminated with the bad news that this drug actually increased the cardiovascular events by 25%, and then increased all-cause mortality by 58%. So it was a disaster, and it's a nightmare created by unintended pharmacological effects. So why this drug failed? There might be two major reasons. The first is the biomarker was probably not causal. So until now, there has been well-established evidence that the HDL cholesterol is probably non-causal. But HDL cholesterol has been strongly and reproducibly associated with lower CHD, cardiovascular health disease risk. But association is not the same as causation. Association just says people with higher HDL tends to have fewer events. But causation says that if we intervene on HDL, if we increase HDL, that can actually reduce cardiovascular events. So association just describes the data, but causation wants to predict if we intervene, then what would happen. So the causation is really what we are more interested in scientific discoveries. And the second reason is that the molecular did something that CETP inhibitor does not. So for example, the molecular actually increased the blood pressure by a lot. So, and also has excess death from sepsis and cancer. So Later CETP inhibitors, there are also additional 3 drugs. So they were found to be neutral or modestly beneficial. Now were not actually reproduced the harm. So this is likely not a CETP target effect of this excess risk. So the harm was off target. That's a property of that molecule, but not of that target. So those 2 questions can both be helped by genetics. So the first question, we can use genetics to distinguish causal biomarkers from correlates. And the second question is that, we can use genetics to help distinguish the target effect from molecular specific effect, from the drug effect that may exhibit off-target effect. So I'll start with the statistical method that we mainly use from using genetics to anchor causal inference. And really the idea is to use the genotype as a natural experiment because genotype are assigned at random, can be viewed as assigned random at conception. And then this is a great device to handle confounding, environmental confounding that's happening. So let's look at this directed acyclic graph, this DAG. So the ZJ will be the genetics, and then X is the exposure of interest, and Y is the outcome of interest. Then X and Y, we are interested in the causal effect of X on Y, but they may be confounded. So there could be a lot of confounding, environmental confounding that we cannot adjust for. So there might be unmeasured confounders that jointly influence X and Y. But the genetics is a great anchor if it satisfies the 3 assumptions that qualify it as an instrumental variable. So the first assumption says that the genetics, it needs to be related with the exposure of interest. And so this means that the genetics needs to probe some variation in the exposure of interest. The second is randomness. So the genetics need to be independent of unmeasured confounder. And this is usually plausible because the genetics are assigned at random at conception. And the third is exclusion restriction, which says there cannot be direct effect from genetics to the outcome. So all the effect of genetics to the outcome must be through the exposure variable. And under these 3 assumptions, then we can easily see how the magic plays here. So the first equation is exposure equation, which relates the exposure to these P genetic variants. And then there could be unmeasured factor, and there can be additional error term. And then the second equation is the outcome equation, which says the causal effect of X on Y is beta 0, and there can be unmeasured confounder in this model as well. Then we know from just standard linear regression course, we know that if we just fit the second equation, we cannot estimate beta 0 because we will have the error that's unmeasured and being correlated with the regress of interest. But then with the first equation, with the help of genetics, then we can plug in equation 1 into equation 2. And then we can immediately see that because the ZJ satisfies those 3 assumptions, then ZJ's effect on the outcome is exactly proportional to its effect on exposure, this little gamma J. So that's great because that says that the genetics effect on the outcome, that's capital gamma J, must be proportional to the effect on the exposure, little gamma J. And the ratio is exactly the causal effect of interest. So this is saying that, with the genetics that's influencing the outcome only through X, if we change X by gamma unit, and then X will change Y by beta unit, then the genetics should change Y by gamma times beta unit. And then we can basically take the ratio and then get the causal effect of interest beta 0. So with that in mind, then we can essentially obtain summary statistics for this genetics association, this little gamma J and capital gamma J. So if we have each independent SNP J, then we just need from the first GWAS, we obtain the SNP exposure association little gamma J hat and the standard error. Standard error. And then the second GWAS, that's obtaining the capital gamma J hat, which is SNP outcome association, and as well as standard error. Then with these summary statistics and no individual level data required, then we can easily solve this equation by just plugging this hat and then take the ratio. Then we can see each of the genetics can calculate the ratio and then estimate the beta 0. But now we have a lot of genetics. So a natural way is just to do a weighted combination of those each genetic specific effect. And then an efficient way to combine them is to do an inverse weighted of the variance. And this gives the natural estimator called inverse variance weighted estimator, so the IVW. So the IVW is just known to be biased when the instruments are weak, which is usually the case in when genetics are the instruments. So the reason is very simple. It's because the denominator, the little gamma j, like when we take the ratio, the little gamma j is measured with error. So the denominator is systematically inflated because the second moment of little gamma j is the signal part and the noise part, the variance part. And then, so this leads to the IVW being attenuated towards the null in the two sample setting. And then as more weak IVs will make this worse. So in an earlier work that we proposed in 2021, 5 years ago, we proposed this debiased IVW that just subtracts the noise and then this corrects the biasing issue and can make this estimator much more robust to weak instruments. And this debiasing technique will be used later as well, but I won't spell out the details. Okay, so that's the device we can use with the genetic instruments. And we know, we have a lot of approaches to estimate the causal effect with the this genetic exposure and genetic outcome associations. So then go back to the drug target question. Then, so we have the device. Then how can we potentially answer this question? So the proteins are the natural exposure to consider because the proteins, they are mutable. They will change after experiencing being on the drug. And they are also measurable, especially the plasma proteins. They are very easily measurable. And they're also the targets of most drugs. And importantly, the proteins, they have cis-pQTLs, which are variants that are known biologically to influence the abundance of the protein. So there's well-known mechanism for these cis-pQTLs to directly influence the protein. So that's more likely to satisfy the exclusion restriction assumption that we mentioned earlier, that the genetic instruments cannot have a direct influence to the outcome, not through the exposure. So the pleiotropy is unlikely, so we have greater confidence that these instruments are valid. And there are also many data sources available for that jointly measure the protein and the genetics, including Decode, ERIC, and UKB. So for these pQTLs, they can now be measured in very high throughput sequencing, including using the SOMAScan and Olink approaches. So now, if we apply these approaches, this mandated randomization that uses genetics to infer the causal effect of this drug target protein, can it do well? So these are some evidence from the literature. It's a mixed effect. There are examples where, for example, this gene, this HMGCR, is known from genetic prediction to lower LDL and fewer cardiovascular events, and there's also a small diabetes signal. So those genetic predictions are all replicated in clinical trial by the statin drug, right? And then there's also the PCSK9, which is well known from genetic study to lower LDL, reduce cardiovascular risk. And this is also shown successfully in drugs that target PCSK9. But there's cases that were not confirmed, such as this IL-6R. So this is found by genetic prediction. It's predicted to lower CHD risk. But there's a very recent trial readout, actually from last month, that showed it's a null result. So this was not confirmed. And then there's also the CETP example I talked earlier, that because the CPT likely have off-target effect. So the target is not the same as molecular. Although the CETP is found to, is genetically predicted to lead to higher HDL, lower LDL, and fewer cardiovascular events, but we saw that torcetrapib actually increased cardiovascular events and increased death risk. So from this result, we know that, the genetic perturbation, they can inform target effect, but a drug may do more than perturb its intended target. So also now with the high throughput sequencing of the proteomics, now we can also identify, just move from known drug target to protein-wide discovery. So the previous approaches works if we know this target, but we can also use the proteomics to discover novel targets. And this is analysis we did and just recently published. So this is using the proteomics from the ERIC and the DECODE study. And because the ERIC study has smaller sample size, so we use a 3-sample design. So we use the ERIC study to select the genetic variants for each of the proteins, and then do the LD clumping to make these genetic instruments independent. And then we use the DECODE, which is another study. And then this is used to estimate the selected SNP and the protein effect. And the prevalent CHD GWAS is obtained from a meta-analysis. That's pretty large meta-analysis from the Aragon 2022 study. So here are just about 10 proteins that are found to be significant from observational studies, and then we wish to see if there's like genetic evidence for them to have causal effect on CHD. So actually, the MMP12, this protein pop out. So this protein has 2 cis-pQTLs that are independent and then are very significantly associated with the MMP12. And this has very strong F statistics indicating that, the genetic instruments are very, very strong. And the MMP12 is found to have an odds ratio of 0.97 on CHD and with a significant p-value. So all the other proteins were not significant. This may be it's not causal. This may be because the genetic study is lack of power. And then there are also some proteins that just lack very strong cis-pQTLs. So for the MMP12, this is actually super interesting because it's found to lower CHD risk. But MMP12 has been found to be associated with increased incident CHD risk just repeated in many studies. So this is a volcano plot, and we see from either the CHS study or the ERIC study. In both studies, the MMP12 is actually found to be quite strongly associated with increased risk of CHD with very, very strong p-value. In contrast, the MR shows that the MMP12 has a very small but protective effect on CHD. So what could be going on? So biologically, either direction may be reasonable. So the MMP12 is an endopeptidase expressed in vascular tissue and atherosclerotic plaque. And this degrades extracellular matrix. This is involved in tissue repair and vascular remodeling. This can reduce inflammation. And there has been previous experimental studies that showed conflicting evidence. Some suggested that it promotes atherosclerosis, and some suggest this may improve healing after ischemia. So the question is, how do we resolve these conflicting results? And is MMP really a cause of vascular disease? Or it may be a marker that's upregulated by vascular disease. So if we do a single exposure mandated randomization, then this cannot tell this apart. Then we need MR to answer more complicated questions. So what we want is essentially a mediation analysis. So mediation analysis lets us to decompose the effect. So the total effect of exposure is the direct effect of the exposure and the indirect effect of the exposure through a mediator. So this plot actually visualizes this, that the exposure may have a direct effect on the outcome and may also have an indirect effect on the outcomes through a mediator. Then the indirect effect can be calculated by just simply multiplication of alpha and beta. And this is a setting where the multivariable Mendelian randomization can be used, because multivariable Mendelian randomization can study the exposure and mediator simultaneously and can find instruments to instrument them both, and therefore can estimate the direct effect of the exposure and the mediator on the outcome. Okay, so that's what we did. It's a multivariable Mendelian randomization. And then we use the CIMT, stands for carotid intima median thickness, to measure the extent of subclinical atherosclerosis. So we conducted a bunch of univariable Mendelian randomization analysis. So first we use MMP12 as the exposure, and we found that, MMP12 actually, so the beta is the log odds, so it's reducing cardiovascular risk. That's what we saw earlier. And then the MMP12 does not seem to causally affect CIMT. So for the CIMT, we also did univariable Mendelian randomization. So the CIMT is found to, significantly increase cardiovascular risk. That's kind of a positive control because we know that CIMT is a risk factor. And then interestingly, the CIMT also significantly increased MMP12. So, and then we also conducted multivariable Mendelian randomization, so with the MMP12 and the CIMT together. And then we find that there are direct effect, the CIMT has a direct effect of increase of 0.13 on CHD, and then the MMP12 also have a small negative effect on CHD from the direct effect. So from a bunch of analysis, this is likely what's going on. So the CIMT is a risk factor, and then it can increase the MMP12 concentration, and then the MMP12 actually has a small protective effect on CHD. So if we just have this piece of information all triangulated together, then we can actually find the decomposition of the total effect of CIMT on the CHD through the MMP12. So the indirect effect is just the multiplication of these two coefficients. It's a very, very small indirect effect. And there is a substantial direct effect of CIMT. Then the total effect can be just calculated, which is very close to the estimated total effect from the univariable MR of CIMT on the CHD. So from this analysis, we can see that, the MMP12 is largely a descendant of subclinical atherosclerosis. And the observational association may be confounded. And this may explain why the observational association is on the opposite direction. And this direct effect of MMP12 is very small, but it's protective. This is also consistent with the biology. So this is an example that I like to show with Mendelian randomization. If we use this in a very clever way, then we can use it to actually answer more complicated questions. So scientifically, this is great, but a statistical question rises. So look at what happens to the instrument when we move from univariable Mendelian randomization to multivariable Mendelian randomization. So with the univariable Mendelian randomization, we only need these instruments to marginally be strongly associated with the exposure. As long as, the instrument, they can perturb, lead to some change in the exposure, that's fine. But for the multivariable Mendelian randomization, we actually need the instrument to have independent genetic signal on each of the exposure in order to distinguish the exposure's effect. So in that case, we need to measure the instrument strength using the conditional F statistic. Basically, this is saying, given one exposure, whether the genetics strongly predicted this other exposure. And then with the two exposures, actually, we can see the conditional F actually decrease a lot. So what happens if we want to consider a lot more exposures simultaneously? So that's really posing statistical challenge. So back to where we started, the lipoproteins, which are essentially the target we want to focus on from the torcetrapid example. And this HDL cholesterol, we know, is just one crude summary of a mass of correlated lipoprotein species with different biology. So with now modern data, we can measure these lipoprotein subfractions, which are like different sizes of subfractions that constitute the HDL cholesterol, LDL cholesterol, etc. And then each of them have GWAS study, so they have GWAS summary statistics. So actually, we can use median randomization potentially to just distinguish the causal effect of these lipoprotein subfractions. But the question, this is scientifically intriguing, but there are questions coming up. So first of all, you can see that this is the genetic association of each of these 27 lipoprotein subfractions. So as you can see, just different subfractions of HDL, and this is a big block of LDL, and so an intermediate density lipoprotein. So these are all very, very highly correlated. So this is an HDL block. This is another HDL block. This is just the low density lipoprotein blocks. So they are very, very correlated. So how can we, so this conditional IV strength, they will collapse as the exposure become highly, highly correlated. So we may not have enough independent genetic signal to distinguish this causal effect of these very highly correlated lipoproteins. And another problem is the interpretability. So if we have a vector of just 27 direct effects among nearly correlated traits, that's also very hard to interpret. And also the inference is challenging. If we do some variable selection, that may be a first idea come to mind. Then this has problem. For example, it has well-known problems such as the lasso will just tend to select an arbitrary exposure out of very correlated exposures, and then how do we do statistical inference after the selection is also a problem. So what can we do then? So we proposed a method called MVMR-PACS that stands for pairwise correlation screening. And this can estimate what genetic can distinguish. So our thought is that with this very highly correlated setting, then it's impossible for genetics, or at least unrealistic for genetics, to distinguish all the causal effect of these highly correlated exposures. Then we will try to use data adaptive way to group this into signal groups, which is defined by either a signal group is a group of exposures that are either having extremely high correlation, or they have the same causal effect. So their effects are indistinguishable. So what we do is, without giving all the formulas and details, what we do is essentially minimizing a debiased objective to remove the weak instrument bias, and we place a pairwise selection loss, which is an extension of group lasso loss. But group lasso needs to pre-specify grouping, but our loss can data adaptively select the grouping. And then we will group the exposure whenever the effects, the data cannot tell apart. And then we will estimate the direct effect of each group. So there is also theoretical guarantee we develop for the variable selection. So how about the post-selection inference? So we use this technique that have been proposed recently called data thinning. So data thinning is a technique that essentially can divide your data into two independent folds. So if we have a single SNP exposure association, we can construct two independent copies by injecting noise to the original association. And then with the two independent copies, we have two independent data sets. So we can use one data set for the selection, and then use another data set for statistical inference that's given the selection. So we developed data thinning for this median randomization setting with genetics summary statistics, and then this can give valid post-selection inference. So this is just a quick show of the results we obtained of these 27 lipoprotein subfractions as well as with traditional lipids. So we can see so these columns, there are 100 100 rows here. They indicate 100 repetitions of the data thinning. The reason is that data thinning has intrinsic randomness. So if we do a data thinning like 100 times, this may give us different grouping. So we instead just plot all the 100 rounds of the data thinning procedures. And then we can see basically the white are insignificant effect and the colored are the significant effect, with the red indicate harmful effect and then the blue indicates protective effect. So we can see that essentially these traditional signals like LDL and triglyceride, they just have very strong harmful effects that are reproduced here. And then we found this block of medium and small sized low density lipoprotein that has protective effect on CHD. And then there's also some like blocks. For example, here, like the medium HDL also is found to have a small protective effect. And then the extra small, very low density lipoprotein has some small harmful effect. So so this is an approach that we can actually find this signal grouping and then identify the direct effect of each of this group. So until now, we have handled the correlated exposures, but in this protein-wide Mendelian randomization study, people routinely analyze related outcomes. But typically the current approach is just analyze one at a time. But that may not be very good, because there are related outcomes. They may have some etiology that share biology. So if we analyze them separately, this does not characterize the shared structure of these multiple related outcomes. And then it can also forfeit efficiency and inflate the multiple testing burden. And this leaves the conditional instruments very, very weak. So our motivating example is this ischemic stroke subtype. There are 3 subtypes, including the large artery, cardioembolic, and small vessel. So if we do a protein-wide MR analysis of these 3 subtypes, each separately, then this has this issue. So our hope is to be able to analyze them together so we can maybe characterize the shared structure and also increase the efficiency. So this is ongoing work from our group that we essentially propose Mendelian randomization with reduced rank structure. So our idea is that, let's say we use the C, this is a matrix, to collect the causal effect of multiple exposures on multiple outcome, where the exposure are all the proteins, outcome are these three stroke subtypes. And then let's suppose if the C is approximately low rank, so the low rank can be understood in the following, that if the exposures, they may affect outcomes through like 2 latent pathways, then this is a rank 2 matrix of C. And then the A matrix will essentially, the A vector will essentially say, A matrix will essentially say how each latent pathway influenced the outcome. So this is a way to characterize the shared biological structure of the multiple outcomes and also try to improve efficiency. So we essentially developed a method with also the debiased objective to remove the weak IV bias, and we have the spectral regularization for addressing the weak instruments. And we propose this reduced rank estimator that's consistent and asymptotically normal under weak instruments. And we also developed a sparse version because a challenge with this analysis, like this kind of latent factor analysis, is that the latent factor can be just a combination of all the exposures, and that can be hard to interpret. Then we developed a sparse version, which will impose each of these latent pathway may be contributed by a small number of the exposures, and that can make the latent pathway more interpretable. So we apply this to the analysis of the proteins and the 3 stroke subtypes. And essentially, we started with the 9 proteins, which are screened by univariable MR, and then with the 3 subtypes. And we have about 177 instruments selected for these 9 proteins. So with our cross-validation procedure, we select a rank of 1. So this means that, this may be a lag power issue, but this means that we select like one common pathway that commonly influencing all the 3 subtypes. There might be other pathways, but there's lack of power to select them in this analysis. So we can see that, this is the our proposed approach. As we can see, these latent pathways contributed densely by all the proteins, but if we impose the sparse version, then this can give us a slightly more interpretable effect in this setting. And then there are some proteins that are, so like the proteins that are most substantial in this pathway are well known to have causal effect on the stroke. So that's good. That's like a positive validation of our results, such as the MMP12 and the HTRA1. So those are well-established protein causally associated with the stroke. And then we apply our method. We can also just give all the point estimates of each of the 9 proteins on the 3 stroke subtypes. So this plot is a lot, so I won't ask you to look at all the details, but we compare with some existing approaches, and these 3 are our proposed approach. So we can see, like in some of these cases, we can see that the reduced rank may be less biased compared to existing approaches, and then it can also have, in some cases, have increased efficiency. Okay. So I also like to talk about the software since we're at the Bioconductor conference. So everything here runs with summary statistics, and the Mendelian randomization is very easy to run. That may explain why Mendelian randomization is just gaining so much popularity. Even people are, there are work calling for like more rigorous Mendelian randomization actually, because they're just so easy to run. So for this, like the IEU GWAS so this and open GWAS and also the GWAS catalog, they have publicly available summary statistics. And then the 2-sample MR and the Mendelian randomization. So these are 2 main packages that actually does the end-to-end Mendelian randomization. So they can start from like SNP pre-processing and then like doing 2-sample Mendelian randomization estimate with diagnosis, etc. So these are all very easy to use. And there are some other packages like the MVMR package that we developed for our multivariable Mendelian randomization approaches. And then we can also have some other packages such as the MR-Presso for outlier detection, and then MR-Reps and the MRC-MR. So those are recent methods that can better handle pleiotropy. And from this talk, the DIVW method is already in the Mendelian randomization package, and then we are developing the MRR and the MVMR PACS package. So those are available now, but it's still under development. Yes, and we will think about, we will work on moving this to bioconductors. So now let me kind of go back to the story I started at the beginning, the torcetrapib story. Since now we talked about like different Mendelian randomization approaches to answer increasingly complicated question. So can we use these Mendelian randomization methods, or like just with genetic anchoring, how can we better handle the torcetrapib cases? So recall that the torcetrapib is to target the CETP genotype, and that reproduce the CETP genotype reproduce the lipid effect, but not the blood pressure. And the on-target inhibition predicts the benefit. The trial actually itself measure the proteomics. So they measure about 1,000 proteins at baseline and also at 3 months after taking the torcetrapib drug. So they also measure this for the control arm. And they found that there are 249 so there are 249 participants on torcetrapib with the proteomics measured. And they found that 200 proteins actually were changed actually with the 3 months of usage of torcetrapib. So that's quite amazing, because with just short-term usage of the protein, we can actually identify a lot of changes through the proteomics, even before the clinical outcome, long before the clinical outcome manifests. And this gives us interesting idea that maybe we can use the change of the proteomics from the usage of the drug to identify off-target effects long before they show clinically. So this is a study we did recently that we want to, so if we do the trial proteomics, then we can see a lot of the protein changes, but we cannot separate the causes from consequences. And then the Mendelian randomization, they can separate cause from consequences, but they can only study the target that we already suspect. They cannot be well used to study the off-target effect with itself. Then we will basically just combine those two. So we start with the 200 proteins that changed in the Illuminate trial. And then there are about 70 protein explained by the lipid change. So these are on-target effects. So these proteins, they are just downstream of the lipid change. And we screen them out, and we are left with 131 off-target proteins. And this, among these 131, there are 95 with cis-pQTLs. And we analyze these 95 proteins against 19 health outcomes that were found to be modified by the torcetrapib in the clinical trial. So this is our analysis results. So basically, these are the 17 of the 95 proteins that have a causal effect on at least one outcome. So these are the 19 outcomes that we study. And then the colored dots are the significant effect. So the blue color indicates the direction is actually discordant with what we would expect from the torcetrapib trial. And then the red indicate the direction that's concordant with what we suspect. So basically, this means that for this protein, we from the Mendelian randomization analysis, we know whether it's increasing or reducing blood pressure. And then from the trial, we see whether this protein is reduced or increased by the drug. Then we can triangulate these two pieces of information to see that whether the induced change in the protein actually from Mendelian randomization actually leads to the expected direction as what we see. So we found that there are 4 proteins. These 4 proteins, they actually show concordant effect with the increased blood pressure that's observed in the trial. So for some of these, they may be reduced by the drug, but they have beneficial effect on, so they can reduce blood pressure. And for some of them, they are increased by the drug, and they were found to increase blood pressure. So those are what we mean by concordant effects. So this indicates that with the proteomics measurement from the trial, and also combined with Mendelian randomization, this may be hopeful to actually identify some off-target effect that from short-term usage of the drug, even long before we observe this clinical outcome. So this can have the benefit of we don't have to expose patients to harmful drugs, for example. So there are, of course, limitations. The estimate is different. So MR is meant to estimate a lifelong perturbation from conception. And the trial is basically estimate just 3 months of a drug. And the tissue is also a problem. So we measure plasma tissue, but it's not the site of action. And if the harm is elsewhere, we may not see it. So there's interesting direction of maybe we can combine tissue-specific MR estimate into those analyses to give us more information. And the assay artifacts is another problem with this affinity-based proteomics measurement. Because a cis variant may alter an epitope, which can affect aptamer binding without changing the concentration. So a cis pQTL may not actually be a cis pQTL that changes concentration. And then the instrument validity. Some proteins, they do not have cis pQTLs. Okay, so that's the end of what I wanted to talk so far. So essentially, I start with the genetic anchoring. Because the genetics, they are assigned random at conception. So that's really a great time zero we can use. And the randomness provides natural experiment for us to like see some causal effect from not only association. And modern Mendelian randomization can address more complicated questions, including mediation, multivariable Mendelian randomization, and the signal groups and low-rank causal structure. And there's also, interesting ideas for translation. So we can use Mendelian randomization and with the proteomics to anticipate drug efficacy and safety very early on. So if we combine proteomics, pQTLs, and outcome GWAS, this can help us prioritize targets and anticipate on or off target effect, even just with early phase trial before phase 3 studies. That's all I wanted to say, and thanks to my collaborators, and thank you for the support from NIGMS, and thanks everyone for listening.
Questions? Any questions?
Hi, thank you. It was great talk. Can you see like applying something like this to like perturb-seq?
Yes, that's a great question. Yeah, I'm very interested in perturb-seq because it's essentially a very similar idea. Like, yeah, we apply some like like inheritation of the genes, and then that's essentially can be viewed as random, and then this can induce like some changes in gene expressions, and then we can use this to, for example, like infer gene regulatory network, etc. So this is very much related to like genetic anchoring idea, because we do have the randomization to anchor with. So there are some works already, and then I'm also working on this topic. Thanks.
I had maybe two questions. One had to do with the causal diagram. You had a few of them, and you might have a node that's a protein, MMP12, let's say.
Yeah.
What exactly is that node? Is that expression of MMP12 or the existence of a variant, non-wild type?
Yeah, that's a great question. So that's the concentration of the protein.
It is. I see. Okay. Then the other question has to do with these pQTLs, which are the SNPs that are shown to associate with expression, right?
Yes.
Now, for any given protein, there are probably a number of these pQTLs in cis, right?
Yes.
How are you summarizing over those? And is there any propagation of the uncertainty of the pQTL effect through the model?
I see. Yeah, that's a very great question too. So, indeed, like in the cis region, so we have a lot of SNPs that are highly correlated, and then essentially we just do like LD clumping to identify independent genetic variants, SNPs in that region, and also have like very strong association with the protein with some p-value thresholding. That's what we do. So like in the MMP12, we identify like 2 SNPs in that cis region, two independent SNPs. And then, so about the genetic, the SNP uncertainty. So there might be like two sources of uncertainty. The first is which SNP we select out of this like highly correlated region of SNPs. So we didn't account for that uncertainty, but that's fine because we essentially do this in two independent data sets. So we use the ERIC study to do the pre-processing to select a SNP. And then we use the decode study to, given the selected SNPs, then we use it to estimate the effect of the SNP on the protein. So all the inference are valid conditioning on the selected SNPs. And so the second uncertainty may be the SNP, the uncertainty in the effect of SNP on the exposure on the protein. So that uncertainty is what we account for, and it's actually one of the reason for a lot of the median randomization methods to have weak instrument bias if it's not accounted for. So we need to explicitly account for that. Thanks.
Yes. Did you look at the haplotype? If you have in the follow-up with the question, if you have cumulative multiple SNP that together that create more effect on the protein expression. So if you look at the haplotype, maybe the association change.
Yeah, that's a very good question. We haven't looked, but yeah, I'd be interested to talk more about this. How do we, what can we do with the appetite information? Mm-hmm. Yeah, thanks.
Hi, great talk. In the genetic anchoring, when you made that assumption that genetics only affect the outcome through the exposure, how reasonable of assumption is that in practice, and what will happen to your estimation if that is not the case?
Yeah, that's a very great question. So yeah, indeed, that's a key assumption. And then I think with the proteomics case, with the cis-pQTL, they are more likely to hold with the SNPs only influencing the protein. They don't have direct influence through other pathway to the outcome. But there could be violations, that's why we have trans-pQTLs. So they may influence other proteins or other pathways. So there are methods, if that assumption is validated, then the estimator, some of these estimators that do not account for it will be biased. But there has been approaches proposed to address this under various additional assumptions. And there are two main types of assumptions. First is to assume maybe the SNPs, the SNP directive, the SNPs that validate assumption may be sparse, there are only a few, then you can use some selection, sparse regularization-based approaches. Or there are another type is to assume that there might be some relationship. So there, the SNP's direct effect on the outcome may be uncorrelated with the intended effect. So they are just two separate pathways, and they are statistically uncorrelated. And then we can have approaches also to handle the direct pathway. So yeah, there are different ways to address violation of that assumption.
Guess I can ask a question. So I'm wondering if your method found, say, a hit or a likely effect, is there a way to follow up on that and try to validate that? And how would you go about doing that?
Yeah. Yeah, that is a great question as well. Yeah, we need to think about validation. So, I mean, some of in the proteomics setting, then it's so some targets are already validated in clinical trials. So those are some are positive. So those are some are existing yeah, so already validated. Some are like prospective. So those are great ways to validate the target. And then, so I think like lab, so wet lab with some cell-based experiments, for example, like perturb-seq. So that may be also a way to validate some of the conclusion. But yeah, I haven't done too much of validation for my work, but yeah, I definitely love to think about this more. Yeah. Thank you.
Any other questions? All right, let's thank the speaker again.
Thank you.